All right, let me see. Yes, okay, that's picking up my voice. That's good. Share the screen. I know. I'll let you guys can kind of chime in on that. They're doing construction of on delivery next door. They're putting the biomanufacturing stuff, right? And program, whatever. But it's loud sometimes, okay? So I usually am gonna have the door closed. It should be unlocked. Even if there's tests going on in here, it should be unlocked. I'm not trying to lock it out on purpose. If it is locked, it's not gonna go on lunch today. We'll talk about test taking policy in a little while. But I want to knock out the Kahoots first. Like I said in the lecture, I use the Kahoots for attendance as well as just to make sure we have either, for lecture, we are reviewing the previous chapter. But in here, these are pre-labs. I know it says on the syllabus that our pre-labs are 4 credits, and we're actually going to be taking the scores for these pre-labs. I'm going to, like 99.9% chance I'm going to be counting these as participation credit, okay? I still say it's just a BS one. This is definitely participation credit, okay? So when we get into the lab safety, we'll have one that's related to lab safety, and we'll get into the exercise one. I expect you guys to come prepared. Now, you're allowed to miss questions here today. You're allowed to do terrible on it. I highly recommend and request, please, to at least like peruse through that lab before you come in and take that test and test the Kahoots. And like I said, it's probably going to be participation credit, as long as I see that you guys are like making an effort to prepare. If I start sensing that like literally you guys are coming in blank every day and the majority of students aren't prepping at all or looking at their labs at all beforehand, or in like trying at all to be prepared for the lab, then I reserve the right, because it says on the syllabus that I'm counting as credit for it, to switch to it being for credit, okay? So try, like I said, if you're mostly making an effort on getting them, I'm probably not going to give a damn. But if I just like everybody's making zeros and we're not even caring at all about the Kahoots in the beginning, then I'm going to switch to making the credit to force us to start preparing for it. Is that fair? Can we kind of do that a little bit? Like I said, just at least like look through the lab and see what we're getting into. It's going to make it a lot easier to get the work done if you have some kind of primer for it. 10 questions typically for our pre-lab Kahoot. Yes, you can use your notes on it. It's not closed notes or something. So if you have your notes, your labs with you, and spoiler alert, every single lab seat has their own microscope next to it. Right down there in that little cubby, microscope. We'll go over lab safety and things like that on the actual lab safety day on our next lab, so don't worry about that right now. But you will have also your own lab manuals. I'm still putting the sheet in the sheet protector. We're almost done with it, but there are a lot more stuff. It's a little bit more effort. But anyways, your lab manual that I have written myself, it has only the information that I want you to learn in the lab. There's no extra. Oh, don't worry about that. We're not going to test over that. This is like edited down to what I need you to know. Okay, but that means you need to know it all. You'll see how it's laid out, like on these lab management, they'll be in your little pocket next to your microscope there. These stay in here. No, you may not check them out, because in case you haven't noticed, if you haven't looked at Canvas, you will have the PDF document for these. It's literally the exact same thing as what's here. Literally, it's what I sent to the print shop. So that's there for you to use on your own time outside of the lab. These stay in here, the physical ones. Want to print it out on your own resources or in the library or something, you can use me. Don't let me stop you. But you don't need to bring anything to this lab as far as that goes. I don't need you to print out your lab exercise. Waste of paper as far as I'm concerned. Okay. You are somebody that likes to have your own lab exercise printed out. Like I said, you can do. But the way we'll be doing things in here is, for example, for exercise 1, literally the first page that you'll open up to is our table of contents, and it'll show you. And you can fold this over so you can handle it nicer in the lab or whatever. But he'll tell you, okay, we're doing exercise 1. That's going to be on page 3. So you can turn page 3, just have right after the lab safety stuff. This is exercise 1. Exercise 1. This is all of the info for the lab on that one page. Every single exercise is going to be. There's very few deviations from that trend. Okay, so all the info that you could be tested over on there, then the protocol over here, is actually following the lab. You can go through and mark it off as you go. Track. Then the next page, this is your study sheet for that one lab. Okay, and then I made it up for you. A lot of it is handwritten, but it's better than not having a study sheet. So there is the study sheet, some of the types of handwritten, and I've made consolidated this info, all of this same exact info. Again. All that same information will be on the first page that I test you over, but consolidated into a nice visually friendly format with pictures, things like that, that we guys are going to study. That's all. Okay. All right. Then we have the actual worksheet, the exercise worksheet. This is what you'll be filling out in lab. So for example, we're not doing these today. When we do start exercise one, which will be on week 3. Okay. Week 3, one of the questions on here. Why do we flame the loop between quadrants of the street plane? We kind of already learned about that in lecture, right? It's to dilute out the cells. I'm not looking for you to write a dang novel on this. If you write out here, spread out cells. Or if you want to write more about it, because it'll help you understand it better, you can write out the cells and separate them individually so we can have individual colonies to create our own pure cultures. Write that. If you write spread out cells, I'm like, yeah, they get it. They know why we're doing it, right? You'll fill this whole thing out with your little answers. We don't have a brain grounds for exercise 1. We'll talk about what that means later on. We'll take that next one. We'll talk more about the lab. But anyway, this is what you'll take the picture of and upload onto your in-lab exercise. Okay. This is the in-lab doc. You literally take a picture of it when we're done in the lab with your phone, upload it onto that exercise. It pops up for me, and I will go through each of these. I will grade them for accuracy, not just participation. You'll get partial credit for participation and partial credit for accuracy. There's a rubric associated with each of these, and based on if you are putting answers that are wrong, but they're in the ballpark, you get partial credit for that. But if you put answers that are just straight up wrong, or you don't answer it, or you don't complete the grand round, you just want to leave early like that, it's your choice, then you get points off for those. Right. Rubric will outline exactly how I will be grading that. And that's going to be associated with that. And when you're done and you've taken your picture, just go take a paper towel and like you'll be squirting down your laptops and everything. We'll be getting nasty every single day. You will wipe it off when you come in and you wipe it off when you can. And then you use that paper towel to wipe this off too. It'll get a little smeary on these because they're not really made for your iris. So if you're having a hard time getting it off or it's real smeary or if less smeary next to you, you can use water or something that is red bottle and sort it on right to get off the filter. Dry erase marker. If you accidentally write on it with a Sharpie, just let me know. I have some alcohol that I'll write on. Your Sharpie and dry erase marker. Right. So that's your lab manual. Like I said, these will stay in here, so you don't need to print for lab exercises. But your lab exercises are all going to be there for you on campus. We'll talk about how that's laid out for you when you start talking about class. I wanted to kind of introduce that to you guys, so you kind of know that, and kind of let you guys get signed in on this. We're going to do the Kahoot now. Everybody signed in and ready for the Kahoot? You know what to expect with this. This is just 5 questions, and they're mostly BS. What? That's okay. All right, this is the scale of 1 to 5. 1 is disagree, strongly disagree. 5 is strongly agree. I am confident. I'm going to use a microscope. Try to spend, I'm recording this and the microphone is on this thing. So I'm going to try to, so you guys like to see how already I migrate around when I'm talking. I'm trying to stay over here so that it records. You have some people that couldn't make it today. Most labs, I won't be like, oh, you're not going to make it today. I'll record it for you. But this is different, right? We're just talking today. So didn't do that for people that couldn't make it, that already notified me and had reasons. All right, so here's our results for our poll. I'm going to do this in the most annoying fashion possible. All right, so most of us are at a 4. All right, I'm not going to call anybody out. For those of you guys who might be rating yourselves at 4 or 5, 99.9%, I believe, I have hard suspicion of this, that those of you who are rating yourselves at 4s and 5s are probably working in vet offices and you're already using microscopes in the vet office.
I've never had a single student who was good at using a microscope. Been here for years. Think about it. So you're probably great at working all the way up to 40x. But when we use the 100x, most people have not used the 100x, and that's pretty much exclusively what we're using in this lab. We're talking oil immersion. If you have never used oil immersion, or you think that's something weird that we almost never use, nobody ever really uses that. BS all the way through, you're using that every day in this lab. And it is hard to get the focus going without somebody guiding you to it. A lot of people that I have too that work in vet offices, somebody's already put the slide together and put it on there for you. And you're just looking at it and looking at the differences. You're doing making your own slides, finding the specimen yourself and focusing yourself. When we get into, that's what the lab is for, is to teach you how to do that. But I think you'll be surprised at how terrible your microscope skills are. And I'm prepared for that, but I want you guys to be prepared for that too. So don't feel bad. The whole reason I'm bringing this up, don't feel bad when you suck at it. Please don't feel bad when you suck at it. Okay. I mean, even had a student last semester. Does anybody in here know who Grace Fisher is? It's okay if you do or don't. That's all right. She is somebody who knows it all. And she started off in my class. She's like, I have a microscope. I use it at home. Didn't know how to use it. Couldn't focus to save her life. Now we got her there, right? That's what the whole point of this class is. But if you suck at it, don't beat yourself up, please. I'm here to help you succeed with that. One of the other things that I like to give a primer of for this part of the course. The lecture is one thing. So I'm talking at you about things, and you're expected to kind of understand it and then use resources to make sure you're understanding it, whatever. Lab is different because you're actually doing things hands-on. You right now are sitting at lab benches with people. I don't really care where you're sitting at right now today, and I don't really care where you sit at next week. But starting week 3, where you sit is where you stay. Because I'm going to assign your table certain microbes, and then your results, I'm going to depend on being for that microbe. So you can't be collecting data from different microbes throughout the semester. We're going to be cataloging it and collecting it throughout the whole semester for your 2 microbes for that table. So you don't want to mess it up with other microbes. So you're going to have assigned seat, not assigned, but you'll be set in your seating chart starting week 3. So think about where you want to be stuck at that point, okay? If you didn't think about it today, start thinking about it maybe next time, because on week 3, I'm writing down your names, okay? Also helps me with learning your names, so I'm actually really bad at that, okay? Anyways, having said that, if you are the type of person who is very much a loner and you do things on your own, and you're able to figure out and problem solve on your own, or you're not afraid to ask for help, you'll probably still be fine. If you work well with groups and you can get along with others, that's great, and that's going to prep you really well for going into the healthcare field. But that's team-based work, not one person against the world. However, if you are a person that you really only ever work on your own, you don't like socializing with others, you're not good at asking for help, and you have been in other lab situations where you felt overwhelmed, but you like barely are scooting through, which is figuring it out on your own, pick up your backbone. I will let you drown. I will let you drown in this class. Because most people who are in my class right now, who are going in a lot, majority, and I know some of you guys are going into vet, congratulations. But if you're in my class in here facing me right now and you're going into nursing, I either have students that are in nursing school right now, but a majority of you guys will be going to nursing either next or once you knock out physio. Those are usually the 2 barriers that people have to cross before going into nursing school. I do not want you to be my nurse or my family's nurse if you can't ask for help, period. I won't let you go. I will stop you. I will be that wall. Because if you can't ask for help, forget it. You don't belong. Drop if you can't. I'm not kidding. If you can't ask for help, drop. Don't go to nursing. You don't belong there. There's going to be plenty of times in your existence in healthcare where you're not going to know what's going on, and you have to know right then. You better go to your charge nurse and ask for help when you don't know. Otherwise, somebody could die. If you can't ask for help in here from somebody that's actually pretty laid back, and I will help you. You come into my class and maybe you're a little late, and you don't get along well with your lab people, but you want to get your work done and whatever, and you just didn't prep that day, whatever. You're just like, I don't know what I'm doing. I just don't even know what exercise. I'm so overwhelmed. And you raise your hand and you call me, Professor Ghost, I don't really know what I'm doing today. Can you catch me up to speed? I will be there and I will help you. And I might even hold your hand through the exercise, but you got to ask first. You've got to ask, okay? If that's a problem for you, you have a hard time asking for help, get over it now. This is the place, okay? I will help you if you ask for help. I will be there every step of the way to help you if you ask for help. But you have to ask first. This is really important. And yes, I have seen people drown because of that. I see people get B's, D's, and F's because they won't ask for help when they had every ability within them to get B's and A's. They just weren't asking for help. Not from me, not from the people they work with. If you're more comfortable asking for help from your lab mates, and they are going to help you and they're capable and they don't mind helping you, that's great too. You're asking for help, and that's fine. Okay, so don't think that's a bad thing. And you guys will get to know each other, you'll get to working together, and you'll get there. But if you don't ask for any help and you're letting yourself down, you do not belong in healthcare, not in vet either, right? You don't be people's pets. My dogs and everything. I don't have kids; they're my kids, and I wouldn't want you taking care of my kids if you can't ask people for help, right? So that's important. So I want to get that out of the way. I know it sounds so severe, but it really is an essential characteristic for you guys. All right, so there's a picture. It's stupid. This is a stupid thing, okay? Which one is me in this picture? 1997, I was in 8th grade. I was 13 years old. Winter dance. Which one's me? No. I forgot about this picture, and then there's a story I'm going to tell you with it here in a second. Yeah, people in general, they're going to get this one, right? So I'm the one in the blue dress with the frizzy long curly hair. That's me. And so this is winter ball and whatever. And so one of the things I want to tell you guys about this, you guys remember how I told you about how there's two groups in eukaryotes, and the two groups there's like the 3 domains. There's eukarya, and then there's the bacteria and the archaea, right? Bacteria, that's whatever. Then archaea, I tell you guys we're not going to talk about them a lot. And we can characterize bacteria, archaea, and eukaryotes basically based on their membrane structures. For the 4 groups for the eukaryotes, one of the groups is called Mendoza cutes. Okay, bear with me. We're going to learn about it in chapter 4, but I want to bring it up now because the picture. Just bear with me. Mendoza cutes, okay? And Mendoza cutes is the category that archaea fall under. And so I think of archaea, sounds like archaic, like old, like really like a long time ago or something like that. And archaea fall into Mendoza cutes. Well, I had a boyfriend in like 7th, 8th grade that I thought he was so cute, and his name was Joey Mendoza. So Mendoza, cute, was a long time ago, it was archaic. And that's literally how I'll tell my students to try to remember that. Anyway, that's Joey Mendoza right there. He was I thought he was a little cutie. Anyways, I forgot that was in that picture, and now when I see it, I'm like, oh yeah, there's a picture of Joey Mendoza that I talk about. I don't know anything about him anymore. All right, so where did I go to undergrad? The picture is kind of a spoiler. Yeah. The girl on the right, and my little sister Erica that I was talking about that I carved Barbies from, the girl on the left. I used to get season tickets to the football games at Stillwater, where I got my undergrad degree. I have a bachelor's degree in biochemistry. I graduated magna cum laude in 2006 with 149 hours under my belt after 4 years, just 4 years, 49 hours. Yeah, I went to school in the summer. I was a try-hard, I'll tell you what. And I was very smart, and I was very successful, and I was very good, which is how I got into the grad school that I went to. But we'll get to that. But yes, I went to OSU Stillwater, biochem. And I still get confused between solute and solvent. It's okay. We all have our Achilles heel. All right, next question is actually for you guys. I'm confident in my note-taking and study skills. Be honest with yourself here. It's kind of why I put some emphasis on note-taking and studying and things like that. You honestly feel like you've been taking classes or you haven't really had to study, like learn study, and all you think it's the way we have to memorize things. Yeah. That's part of it too, right? So here's our poll again. Look at the honesty, you guys. So yeah, and that's okay. I have the module there to help you with note taking, and today I'm actually going to go over how I would expect you to study. Okay, so we'll get some of that in there, and you don't have to use everything that I'm giving. This one's true, false. I got my master's at an Ivy League, studied immunology with a Nobel Prize winner who won the Nobel Prize for COVID vaccine, but is all of that true? Okay. It's okay, it's not for credit. That's me in Philadelphia. next to the Rocky statue by the Philadelphia Museum. That's actually all true. I went to the University of Pennsylvania, which is one of the 8 Ivy sisters. It was ranked #1 in the program that I went into, which was overall cell and molecular biology. And I focused on microbiology, virology, and parasitology. I did very close work, research work, and traveled quite a lot between the two of working with the CDC and the Army at USAMRIID, which is where they do a lot of their classified work. I worked on hemorrhagic fevers, and so classified work, right? So I did a lot of work with them, traveled all over the world, giving talks and presentations and everything as part of my PhD program. I dropped out of that program, not because I wasn't doing well. I was very, very successful, and I was in, success mode 9,000. And then there was a guy. That's a story for another time that I'll say. Okay, that's a story for another time. But so that's how I went up with my master's. They call it a terminal master's degree. I had gotten like halfway through my PhD program, just more than enough to count for a master's degree at that Ivy League institution. When I took advanced immunology as part of my coursework, it was with a man named Drew Weissman, who he and another scientists are the ones that developed the mRNA-based vaccines that are the foundation for the Moderna and Pfizer vaccines for COVID. They just won the Nobel Prize for that a couple of years ago. Pretty impressive. That's who you guys are learning science from. It's actually a pretty good thing for you guys. I do not expect you to rise to that level. A lot of people hear that and they're like, oh, so you're going to expect us to learn all that. No, absolutely not. I've been working in industry long enough. And like I've worked jobs that are hourly pay industry with people who don't even have bachelor's degrees. And that's the level of work that I was accustomed to doing. And I felt like I got lucky with this job. I get paid, like I told you guys, peanuts, but I like the cushiness of the job. And I actually really like the students because you guys have a head on y'all's shoulders. You know how to work. Typically people who have families that they're supporting, or they have a full-time job, or it could be single mothers and all these hardships that you guys are pushing up against to get your degree, as opposed to what we see a lot at the four-year universities of privilege. It's a life that I led. I was a privileged person. I came from family that all had college degrees, and I went to Union High School, very privileged high school. I did well there. I did well at my university, but I tell you what, I learned how to learn when I went to Ivy League. I thought I was smart. Those people really are. They really earned it. And I learned how to learn when I got to that university. I'm hoping I can share that with you guys, the tools that I picked up and how I learned how to work hard. And then I learned how to work hard, really. It's like my hands, my feet working in industry for so long. And now I'm here. Yes, I did get stuck in Oklahoma. Okay, I wouldn't choose to be in Oklahoma, but I actually like teaching here. Like I said, it's not something that I would probably give up for a four-year position if I had the choice. So that's me being honest with you, so you know a little bit about me. Here's a little podium. Not that this really counts for much, but this one maybe more luck than anything. All right. Good job, guys. So we're going to talk a little bit about the class. I'm going to start by with Canvas and just kind of walk them through that. I want this to close. All right. Here we go. All right. So if you go to the Canvas page, we always have our 5 most recent announcements on here. And if I post a new announcement, it'll post over here on the to-do. If you've already read the announcement, you can just click the X, and then it'll bring up things that you actually need to do. And you can narrow that down some. Or as you do them, knock them out and they'll disappear. You guys are not hybrid, so you won't have these hybrid BS over on your side, okay? But it just automatically sets me up as hybrid student because I have all of it. So then you guys will have chapters 1, 2, and 3 on here, and then whatever would follow after that, probably chapter 4, 5, and 7. And then so that'll all be in line of when it's due, and it says the date that it's due as well as the time that it's due by. So this will be Friday by 11:59 p.m. midnight Friday. Okay, so that's when everything for the week is always going to be due. So your lecture chapter quizzes will be due that same Friday midnight. Your in-lab uploads, they're just pictures. Just be sure you've uploaded them by that week by midnight, okay? And then your post-lab quizzes. Post-lab quizzes follow pretty much a similar format as your chapter quizzes. They're timed. So your chapter quizzes, you get 15 minutes to answer 10 multiple choice questions. It's one attempt if you haven't done one yet, right? It's one attempt. So be sure you're ready and you have everything you need to do that. And I'm going to walk you through some tips on how to make that more effective for you in a moment. But anyways, There's the chapter ones. The lab ones are only 5 multiple choice questions, true, false, or multiple choice. And they are 10 minutes. So you get less time, but it's only 5 questions and multiple choice. So again, just be sure you're there to have it ready to take. That'll all be over there. And that's pretty much what you can expect almost every week is those things. And then other things that might be due would be things like projects, which we'll talk about, or exams will also pop up over there, right? Eventually, you'll also see as we get near the end of the semester that you will have things showing up as due, like our pre-lab Kahoots and stuff like that have due dates assigned to it, but you obviously don't submit any grades for this. You just have to show up. I will enter your grades for that. So that's why they don't appear in your modules. But if you go to assignments, it's there, but you just don't submit anything for it. I take care of that. So that's why there aren't any of those greater than signs. So anything you have to submit something for, it'll have those 3 greater than signs in front of it so you can see it easier. What I'm saying about needing to see it easier, if you haven't been to the modules, this is the bread and butter of my hybrid class because they need to access all this stuff. You guys may not. You might not visit the modules very much, but I want you to know what's up with them so you can find the stuff. You've probably been to modules because you have to go through the first up module to open anything else. Almost everybody's done first up, so I'm not too worried about it, okay? But be sure you've knocked that out. It'll unlock everything else. If your shit's still gray on your screen, there's still something you need to mark off for your first up. It'll pop up with little check marks. You should have 4 green check marks. If you don't, you're missing something. It'll have an open circle there, okay? All right, note-taking skills. I'm not gonna go over this in detail, but all of written little documents detailing things you could try for note-taking are on here, and then there's a quiz for that. If you take the quiz, even if you don't get it all right, if you have taken it and taken the time to fill it out, because there's open-ended questions with this, okay, you're supposed to design your own study methods and things like that and answer the questions for it. If you submit that, I will give you 10 out of 10 on it, okay? If you've taken the time to submit it, and that will automatically replace your lowest quiz grade with 10 out of 10. Never had a single student get 10 out of 10 on all the quizzes. Okay, a lot of it is just because they're not reading the choices. Kind of like we had with a couple of the Kahoots on there, where it's like you guys will click at the thing and you're like, oh, I didn't see that one. It's kind of like that with the quizzes too. People are just trying to flick through them. Take the time to read it. Okay? Same with the freaking lecture test too, man. Don't rush. Don't rush yourselves. Also, break the habit of going back and changing your answers. Break that habit. If you're somebody that goes through a test and then you find yourself going over and over and over the questions and you just have a tendency to change your answers a lot, number one, that means you needed to study better before you started. You should be confident in your answers when you go in to take your test. That's number one. That's sign number one that you weren't studying the way that you should. That might not be your fault. The other thing is you're probably not very confident in yourself and your ability to have confidence in your ability to understand these things. That could be an issue too. I don't know how much I can help with that. But the thing is that I have noticed more often than not, your first answer was the correct one. When I go through all the data of the people that are changing their answers, more often than not, the first one was the correct one and you're changing it to the wrong one because you're overthinking it. Okay. So you can break that habit. If you have put an answer and you're going back to change it, make sure you have gone through that process of thinking, why is this the right one and why are the wrong ones before you change it. If you can't do that, don't change it. Okay. All right, weekly things. This is this week's, and week to week it might look different. Obviously, this week we don't really have anything for lab, so it's not what you're going to typically see for lab. But you'll have your overview thing, what you're expected to learn. This week, we have our week 1 study sheet. This is literally what would be on our unit study sheet for the whole unit, but only the parts that apply to this week. That's what that is, okay? The unit study sheet has each week's things all compiled together if you want to work off the big picture sheet, okay? That'll always be towards where we're having our exams. So look at week 4 or something, okay? Anyways, here's week 1's study sheet. This should be your Bible. All right. It starts off with these things. These are our course objectives, the big picture take home messages and what we will be covering for this week. This isn't as important for you. This is more important for me to make sure that I'm covering big picture stuff. What you'll do is if you scroll down, it says individual topics covered. This is for lecture only. These are all of the concepts you should fully understand that could appear on the exams. This is where I'll be pulling from for the exams. But when you guys look at this, you read this, microorganisms need a microscope to be seen. That's a pretty straightforward one. Well, let's scroll down to what we did today, which was chemistry, which I don't know why there's a big space, but there is. Just formatting when I converted it to PDF. So we look down here. Hydrophilic means water loving and usually refers to polar or charged molecules. And that's easy enough. And it's a pretty straightforward statement. And it might just make sense just at face value, and even make a, if you need it, if you weren't 100% sure on the difference between hydrophobic and hydrophilic, make a flashcard on it. If this is one that you like already knew this one, and this is just old hat to you, don't make a flashcard for it. Stop wasting your damn time clicking over through Quizlets that you already know the shit. Stop making flashcards for stuff that you already know. Waste, 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 waste. Never, and in case you didn't hear me say it the first time, never copy word for word your notes. Never copy word for word this. Never copy word for word anything, anything, anything in any course ever. Ever, in my opinion, if you want to actually learn a thing. Now, I guess maybe anatomy, where you have to like learn how to spell the freaking thing correctly, and it has to be this exact spelling and whatever. Maybe then the word for word thing is the way to go. But if you're learning in a course where you're expected to know theory and concept, reword it in your own words. There is gold in that. I'll even prove it in a moment. Anyways, you might look at this. Right. So that might seem simple enough for you, and you think you know it and whatever. So if I were to walk up to you, what's your name? Callie. Callie. Explain that. But you don't have to actually do it. Like, I'm not gonna put you on the spot like that. If you're like, yeah, I probably could, or I don't know if I could, or whatever. If I were to walk up to you and say, explain that to me, it might be an easy, just a couple of words to explain it. Some of these things, though, when you look at some of these things, especially as we go on further on in this stuff, that's going to be the key to helping you succeed. Okay. So if I ask you to explain it, it might just be as simple as rewording it in one word, but it also might be something where, if we look at chapter 3, a good example of the opposite side of this just says 5 eyes, inoculation, incubation, and all that, right? Explain that. Now we're getting into needing more words, right? So this isn't going to give you your definition. This isn't necessarily going to give you the exact word for word what you need to know. I don't do that in my class. Some of these will be like the definitions, like that hydrophobic, hydrophilic one. But this one, this is going to require a little more work on your part to make sense of what that says. And it makes sense we go over it in the lecture. And when I'm talking about incubation, isolation, and all these things, it makes sense when I'm talking about them. But if you can't actually understand it, put it in your own words, you probably aren't going to get it whenever I'm asking questions about the theory behind it on the exam either. You're just expecting me to test you over the exact meaning of these words. I'm not doing that. Okay. Like I said, I might say, hey, there was a nurse who ordered that she thought her patient had staph. So she ordered that a test be done by the lab. The lab chose to use mannitol salt agar salt to inhibit the gram negatives, and then the mannitol part, if you can ferment mannitol, they'll cause a pH change. Which part of that was selective? Which part of that was differential? And which part of that, when they put the swab onto the plate, which step of those 5 eyes did that fall? When she put the plate into the incubator, which step of the 5 eyes was that? When she did the streak plate for it, which, you know, so be able to apply these concepts. Don't just be able to recognize that exact word. So there's that. All right. So I'll walk you through some ideas how to get that down. So maybe you're not sure how to get that down, and that's fine. We'll get there. So that's our overview of, then, like I said, that your study sheet, that's going to be your bread and butter, and I'm going to show you how to use it in a little bit. For the lab, today I gave you the link to the whole lab manual. But when we get into the actual lab exercises, I'll give you a link to lab exercise 1 and lab exercise 2 for week 3, for example. So have the individual ones, they're the same ones that are in the lab manual, but I'll just give you those exercises. So when you do the quizzes, you just have that one there. You know, you don't have to find it in the whole lab manual and all that. So it'll break it down for you. But today, hey, here's a link to the whole thing in case you wanted it. All right. Whatever. So preview, like I've told you guys before, whatever I have under preview, it even says on it, not for credit. Not for credit. You do not have to do these things. This is stuff that I have in preview because I'm suggesting that you do it before your lecture or your lab to kind of prime you for it. If this isn't a thing that you like doing before the lecture or lab, maybe you want to do something else, that's great. Again, I have other suggestions for you. For example, you could do one of these. These are the connect homeworks where you have to like click through the thing to master it. You don't have to complete them at all. You could just like not do them all together and you'd be fine. You can get 100% in my class without doing that. So you don't have to do it. It's there for a resource for you. Or you could just do like a few questions. You got bored of it. You didn't want to finish it. So these take like 30 minutes to get through. Okay. I can't change that. That's just how it's formatted. They also have more information than you have to learn in my class. Go off of what I give you in that study sheet, not off of what's in that. Because I can't change what it gives you from the textbook. It just gives you everything that it has in the whole textbook. Okay. I can't edit it out. So I don't make those. McGraw-Hill makes them. That's why I don't like them. That's why I have my quizzes. I wrote my quizzes. They're only going to be over things that are going to be tested. Okay. So keep that in mind. But it's there for a resource if you want to use it, if you know better how to weed out the things that you don't need to care about. Right? There's not a lot, but occasionally there might be something. I had a student email me saying, hey, I wrote my own study questions about this, and they had questions about some topics. It was like, I don't even know what it is because I don't teach it. It was a couple of topics that are in one of those, chapter 1 or 3, that we don't have to know for this class, but it's in the textbook. And I'm just like, that's the danger. That's why I want you guys to stick to that study sheet. Don't waste your time if it's not on the study sheet. Okay. I know some of those concepts are big picture concepts. If you're ever like starting to realize that maybe you're deviating from that study sheet and you're not sure if you need to study a thing, you can always email me and verify it. If you start getting into that territory and you're not sure. Okay. But odds are, if I don't talk about it in the lecture, you don't even know it. I'm not gonna bullshit that I don't talk about in my own lectures for your exams. That's bullshit. So I know other professors do that. I'm not going to be doing it. Be sure you understand what I am talking about. So those are there for you to prep. Another thing that's a great tool to help you to prep with is Sharpen. If you guys don't know what Sharpen is, it's an app from McGraw-Hill, and you can search for our textbook. And I can never remember the name of our textbook, but it is on our syllabus, and it is on the McGraw-Hill site. Okay. And it's literally the name of the textbook you can search for microbiology. It'll give you all of their microbiology. There's like 3 microbiology textbooks they have. Just pick the one that's for our course based on our syllabus and on McGraw-Hill. And it's free. It is. But I'll say this, it wants you to subscribe to like all of their content, and it is expensive. Don't. Just keep with the free version. Promise. Okay. But anyway, the free version of Sharpen, if you do download it, it's for phones, like for Android and for iPhone. It has summaries of the chapters that are like 3 to 10 minute long summaries, and they're typed out. So you could read the summaries, or you can play it and it'll like speak out the summary to you. It'll read it for you. And so it'll play like 3 to 5 minutes of like the summary of what that chapter is going to be about. You could listen to that on like some free time to prep for the lecture, or like while you're waiting for me to come into class, just listen to that and be like, oh, okay. Oh, she's going to talk about this. I have no idea what that is. Maybe go look up a term or something like that, kind of prep yourself for the thing and whatever. So if you want to prep for the lectures, because I know I talk fast and it doesn't get easier from here, that's another tool you can use. You can use chapter homework, you can use Sharpen, or you can like look at the study sheet and just pick out the topics that you just are total Greek to you and prep yourself however you want that way. So if you're a prep before the lecture person, prep before the lecture. Plenty of resources. If you need more, just hit me up and I'll help you. For the lab, eventually we will have the virtual labs on here. Again, not for credit, but if you want to do a virtual lab so that you're a little bit prepped for the actual Kahoots before you come in, they don't take very long to go through the virtual labs. And it's literally like walk through mimicking like what your hands-on experience is going to be there. So it'll prep you some. It's one way to do it that it kind of does work for you. Anyways, now on to the part that actually requires you to be involved in it, like requires it, is the lecture. Obviously, I have the Zoom link, so if you guys ever need to join in via Zoom, I also have the times that you could join in. So if you can't, just someday you have some doctor's appointment or something at 10 a.m., you can't make it to our 10 a.m. lecture, maybe the day before on Monday or Wednesday, you can do their 10 a.m. lecture and join in live. Or of course, you can always watch the recordings that I'm trying to keep up with posting. It doesn't help when I don't have internet. I literally didn't have internet yesterday in class. And so like halfway through class, I started my recording. I can't help that, but yeah, so you should be able to join in live most of the time at those times. Or if you miss a lecture and you just want to be there in person, then I can always tell you guys what room we're having a lecture if you need to go to a different lecture or something like that. Okay, so that's there. We have the week 1 current lecture recordings immediately after that I will update throughout the week. So you'll be able to click on this and find the lecture recordings as I'm posting them on here. Right, and it's like this one, I didn't have internet, so it's only a partial. You guys, this will be the full thing. So there's that. But that's always going to be there if you want to rewatch a lecture. Everybody else is learning the same thing as you, but some people just like to watch what would happen in their own classroom, and that's there. All right. There's recordings for that. And then there's the lecture content. Chapter 1's not a good example of it, so I'm gonna go down to chapter 2. We did today. You click on these are mostly for my online people. They are pre-recorded from past. You can see my hair. I clearly don't have long hair anymore. These are old. They're from past semesters. They might have more info, never less, but they might have more info than what you need to know. I've only cut back. I've never added on content. So it might have more. So be sure that you, like it says in this, it's just basically like just warning you guys. Follow your study sheet when you're looking at that, okay? Those are old. The current ones are current. But there's also a summary of the chapter down here too. So if you wanted to read this before the lab, you could read this before and then come to the lecture, and it has that rundown. This is cut way back on like what we were learning. So that's there too. That's in the lecture content. Come on, dude. Keep up. No. Right. So lecture content. And then under lecture content, we have the Kahoots. So we have chapter 1 and chapter 3. We only did chapter 3 today. So if you wanted on your own time to go back to chapter 1 for your own review and study methods, that's there for you. Okay. We're not going to do chapter 1 in our class. But typically, like when we come back on next Thursday after the break, because we don't have class on Tuesday, to keep you guys on track with my Monday, Wednesday class. Okay. We do not have class on Tuesday. We have class on Thursday. We will start chapter 4. Before we do chapter 4, we'll have the chapter 2 review Kahoot. That's literally this exact same Kahoot. It's just we'll be doing it in class. These will let you go through individually. If you've already done the Kahoot, you can still retake these. You'll just need a new username. I'm pretty sure you can just retake these as many times as long as you are just doing a different username. So be sure you're doing that. So there's that. My hybrid people have to submit that as evidence. You guys, we do it in class, and that's your evidence that you participated in it. They have to do it on their own time and submit a picture of it. Again, it's participation based. They don't get graded for it. Just like you. Then we have audio shorts. These are literally just snippets of main concepts. So here, if you weren't sure what germ theory was, here this is talking about germ theory is what Pasteur came up with to replace spontaneous generation. So that's talking about that. 7 minutes of me talking about that. 7 minutes of me talking about selective and differential media. Microscopy, chemical bonds, pH, 9 minutes of that. So little snippets of topics that if you are having a hard time with something, maybe the stuff we've done so far isn't hard for you, but as we're going on, you aren't entirely clear on the difference between gram-positive and gram-negative bacteria. This is a bread and butter concept when we get into microbiology. You don't get this, I promise you will by the end of the course. Only we drill it into your freaking head so hard, you're not gonna be able to get away with it from it. But this is 9 minutes of talking about that. So if you ever needed to come to something that you're having a hard time with, you could find those things and listen to me talk about it for 10 minutes or less. Okay. Then under that, we will have a link to the weekly reviews. I sent out a notice about the weekly reviews, 10 a.m. on Friday, every single Friday, unless I say otherwise in the announcements. If I'm going on a trip or something, right? Or if I have a doctor's appointment, I will let you know. And basically, it's the same Zoom link that I always use. And so you'll be able to just get on that same link at 10 a.m. on Friday and join in, and I'll be doing it via Zoom. I will cover the major topics that we went over, and then I will cover the most missed topics in a little more detail. I'll give examples of questions. No, they won't be the exact same questions as what's on the exam, so don't expect that. There's no secret like that in my class, okay? There really isn't. I don't know if you guys, any of you guys take chemistry with either Dr. Tim or Dr. Singh. Yeah, all of their test questions came directly from their quizzes. 100%.
It's very obvious.
Yeah.
Not the wording's not.
If you didn't figure it out, yeah. Yeah, it's the same. My class is not that, so don't expect it. I'm being transparent here, so you're not trying to find the cheat. No, there really isn't one. Okay, I've already told you there might be a handful of questions that I did because I did get a little lazy, but a handful. Okay, so be prepared for that. But this weekly review, like I said, I'm not going to give away, so you're not missing that. I literally had a calculus professor that if you went to his reviews, it was all of the questions that were on the test and all the answers and how to work them out. And we were allowed to bring a note card to class and write whatever we wanted on it. So I just wrote the whole test out. And he got fired the next semester, but I got my A. So, hey, that might be why part of the reason why I'm so bad at math, because nobody ever actually taught it to me, but yeah, I feel like I've barely been squeaking by on math, you guys. I'm terrible at math. So anyways, this Go to weekly reviews if you can at 10 a.m. It is on Zoom. I will record it and post it, even if nobody shows up. All right. So you can still benefit from that because I know people have jobs or they're just tired and they can't make it or whatever. And I understand. You're willing to still take the time and watch the recording. Like that's good on you, man. You're doing the effort. So I will still record it and I will still post it. You better believe that you're not going to see me on the camera on that. I'm going to be in my pajamas in my bed probably. When the weather gets nice, I'll be outside on my little swing chair. You'll be hearing birds chirping and the chimes in the background. So yeah, I'm going to be there. I'm going to be talking about my stuff and I'll be prepared for it. But this is just a review. Like I try to keep it at an hour, but you know how I can talk already. I'm sure you can tell. So there's that. In the lab section, we don't have anything this week, but eventually this is where you will see links for your in-lab uploads. That's really all that's going to be there for the labs. Also in the learning areas where we'll have our projects. We have 2 projects for the whole course, one for lecture and one for lab. Just one for lecture and just one for lab. The lab project, we are going to start that on week 4, exercise 3, whatever week that is. We'll start collecting data for our lab project. And from exercise 3 forward, you have only those microbes that your table will be working with. And so that's why it's important that we have our tables established by then. Anyways, we'll be collecting data throughout the entire semester, and we need to keep that data, whether you were positive for this test or negative for that test. And I'll help you walk through and make sure we're keeping that data. But there's a form for that, and when we get to it, I'll make sure everybody knows where it is and how to fill it out and everything like that. And I will walk you through it as we go so that there's no confusion about it. But we'll be putting in here basically what you were positive for and what you are negative for all of the stains and all the different differential and selective media. It's actually coming, y'all. You're actually going to be using that stuff. So all your results for all the tests we're doing for the whole semester. And then after you collected all of that data, I will give you a key that you'll be able to use to determine what your organisms are. I will verify if you're right or not. I'm going to be in the dark on that. We'll help figure that out together. And then for your 2 organisms, you'll answer some questions for those 2 organisms, the same questions for both of them. And then on the last question is, I want you guys to describe to me how 2 of the tests that we did in the second half of the semester worked. That's the whole project for the lab project. It is work that we're going to be doing the whole semester, so it shouldn't be all at once work for you if you're keeping up with it. And I will help you through it. But that's the lab unknown project, okay? For the lecture project, I haven't posted anything as far as what you're assigned. And right now, if you were to look on Canvas, you're assigned Unit 2 and 3 and 4 projects. But that will change once I have finalized my roster. We still have people adding and dropping the class for a little while. Once people are done adding, I think they have until the end of this week to finish that out, or maybe next week, I don't really know. But once that's done, after next week probably, I will post what unit you will be presenting on and what project you will be presenting on. So specifically what topic you'll get in that unit. It's going to be alphabetical, just a heads up. So if your last name is an A, you're probably going to get stuck in unit 2. If your last name is a Z, then you're probably going to get stuck in unit 4. You get me? So that's how it's going to be divided up. We'll have 8 people roughly. This is a full class. There's supposed to be everybody here. But when we get to that, so it'll be 8 people per unit, and you'll be assigned your one topic, and you're the only one that's presenting on that topic. And it's your responsibility to review your peers on that topic. So for example, for unit 2, the first topic that's on that list is, so just a heads up, I think who is it? I have the list, of course, I had the list of people here of that, and I put it all away. Anybody know who's the first to the list? I'm sure if you are the first to the list, you probably know. Lacey. So you're gonna probably, odds are, unless we have some other person with AA or something, because your last name is A there, you are probably going to be the first one on the list. And the first topic that we have for unit 2 is DNA replication. So that's probably going to be your topic. So what she would do is she put together slides or a Kahoot or something else, read on the project, like what you can do anything you want to review. A quiz, do a mini lecture. You could make literally like old school, like poster board, whatever you want to do to review on the topic. But you have to cover everything that we had to learn. So that all of that should be in your Kahoot or your slides or your project, whatever your poster board, whatever it is. And you only have 5 to 8 minutes to summarize. So it's more important to get all of the info in your actual slides or whatever You have 5 to 8 minutes to kind of summarize that. You don't have to cover every detail in your actual presentation. Just give us a rundown of how DNA replication work. And then it's all contained in there. And then we would disseminate that to the whole class if they wanted to use that for their own study purpose. But man, guaranteed that probably when you go take the Unit 2 test, you're probably going to be set on DNA replication. You're going to be an expert on that topic. You're going to have to explain it to the rest of the class. And so that'll be what we do on our review days. That's the class period prior to the exam. So we'll have 8 major topics. Obviously, there's a lot more to the class than just those 8 topics for each unit. But 8 major topics. You'll be assigned one of those major topics. You'll put all the info you can that we have to know about in those. You can use whatever resources you want to, pictures, information from other textbooks, or from peer-reviewed journals, or from just like, Amoeba Sisters videos, or whatever it is. And then you're going to explain the summary of it to me. And then you have to present those in person in front of the class. Or you can record it. I think I've said this before, right? You can record it, but I will play it in front of the whole class. So we'll get in front of the whole class. Like I said, personally, I'd rather just talk in front of people and not see my face and my voice up on a screen. That would be a nightmare for me. I hate recordings of my own voice. But it is up to you, but it will get played in front of everybody else. So that's what that's going to be. For Unit 1, because I know there's going to be darkness about like, so how, like what exactly is expected and whatever. For Unit 1, I'm covering 8 topics myself. I'll try to use different formats so you guys can see some variety in there. It's going to be what I would expect of you guys. All right. And I'll post that in the discussion section so you guys will have that for reference when your thing comes up. You may switch with anybody you want. So if Lacey, right, that was what your name was. If she doesn't want to go on the very, very first one, she can find one of you that does want to just get it over with, or wants that particular topic because they understand it well, or whatever. You find somebody you want to switch with and they agree to switch with you, you come verify it with me first, that I know to change my list, verify it with me, and then you can switch. You may only switch one time, and you have until Unit 2 to figure it out. So once Unit 2 presentations show up, that due date comes, which is the midnight before, then you had better have your switches done. Okay, I will post what your topic is and what days and everything within the next couple of weeks, probably. Okay, that's the project for that. That's coming. Everybody has to do one. It's worth a pretty big chunk of your grade too. I think it's like 10% of your grade, just for that 5-minute thing over that one topic. So it should be easy point, right? In the past, I had groups and you had a project for every single unit. And man, are y'all not ready for some group work to get graded as a group? I mean, it would be like one person might carry the group. I don't know if you guys know that, but that is like real life.
Yeah.
People are not mature enough for that, turns out. So I don't want to talk about that again this semester. People crying and screaming at each other in my office. I'm not doing that again. So I made the rules. So yeah. But we're not doing that again. So it's individual again. We only have to do one project because that's what I can fit into time. Okay. But at least, hey, I'm just gonna get you guys up there and you'll have to speak in front of other people. It's just the one time though. All right. And then we have the exams. I think we're at that point, right? We talked about the quizzes. So I'm gonna tell you something about the quizzes. Yeah. Obviously you get 15 minutes and you get the one try and it's all multiple choice. But what I would do if I were you guys and I was prepping for the quizzes, yeah, have your notes there. That's a great start. Maybe open the PowerPoint and or the study sheet. But there's one more trick to this that I feel like people forget about, and it's very easy little trick. Just really quick. Be sure, like the slides that you can find on Canvas, you can open them within Canvas, obviously, but download them, okay? And open them as a PowerPoint file. All right, we've got here PowerPoint file of, this is chapter 1. Control F. Let's say you have a question on your exam that's asking about inoculation and you're not really sure what inoculation was. And you don't want to futz around with your notes or whatever. Inoculation. Stop messing around with scrolling through your slides. Waste of your time. Use the Control F. Use the Control F. Oh my God. I know it seems like some of you guys have probably been using it. You're like, well, duh. But then some of you guys have probably been scrolling through your damn slides. Stop that. It's okay. Pull it up in PowerPoint and Control F and search for the term that's about your quiz. Seems so obvious and so simple now, right? Don't you wish you'd been doing it? All right, so there's that. It's open note. It's open note. Open your notes. Open the slides and look for it. It's probably right there in the slides, okay? So you have that, but you still have to be able to relate the information to the question, and that's the more important part with the quizzes, okay? And that's why I time it, because I want you guys to be able to at least have had some preface to understanding that, so that you can apply it in a way that makes sense. Most people take 5 minutes or less on those quizzes, man. They really shouldn't take you the 15 minutes. So there's that. There's that. Guess what else you can do? You can open up any of those PDF files, whether it is the study sheets or the PDF of the lab manuals or the individual lab exercises that I have. Open up lab exercise 1 whenever I give that to you, and Control F in the PDF document, and you can search within that too for your lab quiz. Do it. Okay, that'll help you. You're welcome. Seems easy. I don't want you guys to forget about easy tools like that. So that'll help you with those quizzes. With that. Last thing, I'm going to talk to you a little bit about how to study. We've got 15 minutes left. We got 15. These clocks, by the way, are fast. So we'll always go by like watch times and phone times. So if you think that you get in here and you look at these clocks, you're like, oh shit, I'm late to class.
It's almost exactly 2 minutes late.
Yeah, this one's 2, but that one is like on another round. So it's like a whole of 5 minutes or something fast, if not more. So anyways, they're good for like if you need to have your stain sit for 2 minutes, you can use that. That'll be fine for that. But yeah, whatever. So for studying for the class, I do recommend prepping a little bit for the lectures. Like I said, do some sort of summary thing on Sharpen, or read through the little quick thing on the lecture content online, whatever. It just kind of helps get you in that thinking of what we're going to be talking about for that class. So that's something, it won't feel like Greek as much, especially when we get into these next chapters. It's going to hit you when we get into chapter 4. This chapter 4, we're learning about bacteria. Y'all don't know a damn thing about some bacteria. And so, especially if I'm already sitting here talking about peptidoglycan, gram positive, gram negative, I'm going to be bringing those terms up almost every single day after that point. So you need to keep up with that. And it's just going to get more and more and more. So prepping will help you a little bit for that. I'm not going to force that on you though. And I have on the board kind of the layout of the course, the actual, like what you're doing in lecture. That's what we're doing. In lab, that's what you're doing. So we can keep up with that. But prep for the lecture if you want to. I gave you the resources for it, whatever. My recommendation when you are taking notes, use the study sheet. And that's why I say it's your Bible, right? I recommend getting like a Word document, or I use Good Notes. So if you guys don't know what Good Notes is, it's like $10 a year for a subscription to it. And you can use it on your computer. You can use it on your tablet. You can use it on your phones. And it doesn't matter if they have it for Android, they have it for Apple. I feel like Notes is not using it. I'm going to show you how I use it in a moment. But I like Good Notes. So it's like digital notes, right? And you can handwrite and you can convert it into actual typed text. It'll do it for you. And so it's just like, it's so good. You can cut and paste and put things in, and it's so good. But I would, whether you're doing it by hand or digitally or whatever, take the study sheet, and I would take each point as we're going through it in the lecture and clarify it in my own words and with whatever extra verbiage or explanation that I need for each point, and make sure that as we're going through the lecture, I'm understanding each topic that's on the study sheet. That's how I would write my notes. That's how I would write my notes if it was me. Okay. If you want to just take notes from the actual PowerPoint slide, that's fine too. But then I would make sure that I go back to the study sheet and I get all those topics covered. Okay. This is pretty much exactly how your nursing school is going to go, by the way. They're going to give you outcomes that you have to meet. And what people will tell you, if you don't want to drown in all that, you need to be following the outcome. And that's pretty much what my study sheets are like. Okay. They're equivalent to that. So follow that. Make sure you understand each topic that's on those study sheets. Take whatever notes you need to help you understand that stuff. Pull that from the slides, take notes in the slides, and then reiterate it back, whatever works best for you. That's what I would recommend doing. There are terms, of course, that you might want to just use for flashcards and memorize, and that's fine too. It has a place. Okay. What I would do is take my notes in the class based on the study sheet. And this little pink thing that I have drawn on the board, this is a method of note-taking called the Cornell method of notes. All right, and then you can see even on Good Notes, they have a sheet that is like that, that's set up like that. And what you're supposed to do is, what I'm supposed to do, that's all. I'd known about this when I was in school. So in your notes area, this is where I would have my study sheet points, and I would be writing out what I need to write in order to clarify each topic as I went in the notes section. Out here on this left bar over here, you would write big terms or major concepts. You can do it as you go, or you can do it afterwards. And at the bottom, for each page of notes that you have, a summary of what's on that page, just summarize it. I do that either after or during. Also, whatever works best for you. This is like a proven way to help organize your study notes and make sure that you're actually getting the big picture and doing stuff out, not just writing words down. That's the Cornell method of note taking, if you want to do it. Either way, make sure that you are understanding the topics as we're going through the lecture. And at the very least, making a note of, I don't quite understand this, I'm going to come back to this later. Ask ChatGPT to explain it to you. Or look it up in the textbook, or talk to one of your peers, or work in a study group. Make sure you're understanding all of those topics as well as you can, so that if I were to ask you to explain each of the topics, you could. Right? That's how well you want to understand. All right, then. So that's the idea of taking the notes during it. Then I make flashcards of big topics that are new to you, or terms as we're going through them, right? Like peptidoglycan, you might want a flashcard for that one, okay? So things like that. Make your flashcards for that. They do have their place. And then what I would do after each lecture, if I take my notes, you can have your organization of your notes as you go, or I would take time afterward. And then I would either make my terms and my summaries afterwards, or just go straight into my note reorganization, which I'm going to show you. Then, also at that same time, when I'm reorganizing my notes and restructuring and reformatting them in my own words and in a nice organized format, because I know how messy notes can get during lecture. Okay? Once I have like reformatted my notes, I'd also at the same time be making my flashcards for the terms. But again, only the ones that you need. If you already knew the term, stop making a flashcard. But just because it's a term doesn't mean you need to write it down. You already knew it. You don't need to make a flashcard on it. Right? If I could come up to you and be like, what does hydrophobic mean? And you know, oh, that's water fearing. That's probably going to be the ones that are non-polar, because water's polar. They won't mix well. You can say that to me. You don't need a damn flashcard for that. Okay? Just forget it. You already know it. So make a flashcard for what you need. Then I would use the Lightner method to study it, because it really does help cut down on time you're spending. Yep. Pile 1. This is like the pile that they all go in once you make them first. If you can see a flashcard 3 times and get it right, then it goes in pile 2. Right? Or if you see it and you just know that you know how somebody can get their flashcard and I know that one, put it in pile 2. Okay, it probably doesn't need to be there, but if you still want to keep it there just in case, then put it in pile 2. Okay. Once you get your flashcards in pile 2, I would try to get all of them in pile 2 that first day when you first made them, just to knock it out of the way, right? Come back to pile 2 a couple days later. My recommendation, leave it for a couple days, then come back to pile 2, go through them again. Once they get into pile 3, I would leave them there personally until the test. Our tests are only like every 4 weeks. That's not very long. So you can come back to that pile every week if you want to make sure you're up with that. If you're one of those people that likes to be on top of those things. Me personally, if I got my pile moved to pile 3, I'd leave it there to the test. So I would like make my pile, make sure I learned it. It stayed in pile 2. Couple days to a week later, go through pile 2, make sure I still understand it, goes to pile 3. If it makes it to pile 3, if I didn't get it again, let's say I'm going through pile 2 and I didn't know a term. I get it wrong, guess where it goes? 1. It don't stay in 2. It goes to 1. And you better come back to that the next day. I mean it, like the next day. The next chapter that you're adding new cards to that, it goes back on that one that you didn't get right the last time. And you're making sure you get it into pile 2 again. And this next time, it better be going to 3. If it doesn't go to 3, guess where it's going again? 1. Okay. So once it gets into 3, though, I personally would leave it there. Personally. Okay. Then on test day, my reformatted notes, which I'll show you how I would reformat my notes. I would just make sure I could look through that and it all made sense. If I found something that still didn't make sense to me, I would do the work. Make sure it made sense to me again. If it should have made sense to me when it made it onto that list, onto that reformatted notes. Nothing should be reformatted on your notes, reorganizing your notes, restructuring. Nothing should be reformatted. That make sure you understand it first. Everything should be understood at that point. Then when you go through that, just look over it, make sure it still makes sense to you. Brush up on the things that are a little rusty. Go through your pile that's in pile 3 just one more time, or 3 if you're just really that person. You shouldn't be going through more than that. That's what I would do before my exam. My sheets that I made for all those 6 chapters and all those flashcards, just go through them one more time. I ain't cramming tonight for what a waste of your energy. What a waste of your brain. You're just killing yourself. It's your own fault for putting yourself through that. Tell me that all y'all haven't done that. All of y'all been. waiting the night before staying up hours studying and studying and studying. You're probably not getting a damn thing in your brain about any of the topics either. You actually learn that stuff when you do that. You might be able to pass the test. That's not the same thing.
Memorization is
Right. So I'm trying to help you here to learn the stuff. And I know when you hear me talking about this, taking an hour after class to reorganize your notes, and that's what it should take you if you're taking good notes. If you're taking notes to explain those topics on your study sheet, and you're actually explaining them in your words to make sense of them, then reformatting your notes in an hour. Do that then, so that when the day before the test comes, all you do is just read over that sheet and flip through your cards once. You're good. And guaranteed you'll be better prepared for that exam than you've ever been prepared for any of those other exams you crammed for the night before. Guaranteed. Okay, let me show you how I would reorganize my notes. This is for lecture. I'm gonna pass it around. Please pass it around. Everybody take a look at it. If you want to take a picture of it, if you can. And that provides some of these throughout the semester to remind you guys that this is chapter 2. This is what we lectured on today. This is how I formatted my notes for this in the Good Notes app. It's all handwritten. So if you are a handwriting notes person, this is how you might format it on your papers. If you want to use Good Notes, you can do it with by handwriting in the Good Notes, or you can use it by typing, whatever you like to do. But it's like I have written my notes out on one page. Every single thing that's on every topic for your study sheet is all on that one page for that chapter. One page per chapter. If you want to do one page front and back, that's fine too. But I would just keep it on one sheet like that if I were you. One sheet for each chapter. This is just one page for me. And that is every topic that is on your study sheet. And it seems easy enough to look at it that way. But be sure it's in your words and then just explain each of those topics well enough that you understand it, that you could explain it to me if you had to. Some of that stuff I might just have a basic like definition for. Things like hydrophobic, I might have it on there. Well, I know what it means. I don't need a whole freaking novel written out for you. But there might be other things you need more written. Make sure you're writing what you need. It's sectioned out. It might not be in the prettiest sectioning ever. You can redo yours to make it as pretty as you want to, or as clearly defined as you want to. Put pictures. Cut and paste pictures out of the lecture and put it in your Good Notes, because you can literally upload the PDFs into Good Notes, and hopefully it'll rotate that in. Yeah. You can upload your PDF or your PowerPoint or whatever it is into Good Notes, and then you can like circle things and cut it and put it into this thing, and then write your own notes about it. That's what I think makes it like a fantastic app, and you'll kind of see what I mean, hopefully. But if this is something that is helpful to you, I recommend writing your notes about your study sheet, and clarifying that, and then going through that and making it a nice, clear, concise page. However you like to format that. Doing that alone, just making one of these pages, is probably more than enough to prep you for that chapter. That's why I say you make the page. The only time you come back to that is when you're studying for the exam. When I say study, you're going to look over that and make sure it still makes sense to you. There might be a topic on there that it's been a while. What was that again? You need to go back to your notes and revisit it. You can, but odds are just by making that and putting it in your own words, you know. Okay, so that's my recommendation. So that's what I call in my set notes taking thing. That's what I call the bare minimum. Okay. That's what I call the bare minimum. You want to do well in those classes, you really want to excel, make sure you're getting yourself that A, or bring up your GPA, whatever. That's quite an achievement in this class. I'm not saying you'll do all the time. I'm just saying, like, it does take work to get an A, right? So if you want to be that person, make that, and then explain the test, explain the sheet to somebody, to your wall, to your steering wheel, to your cat, to your kid, to your husband, to your coworker. Explain the sheet to them. And how I would do that is as follows. So, what's your name? Tia. Okay, this is Tia. So if it was me and this was my sheet and she was my study partner, I'd be like, okay, Tia, this is my study sheet and I'm reading for chapter 2. So in this section up here, I have like my chemical bonds and my pH stuff. Chemical bonds, the ones that we talked about in class, with the covalent, the ionic, and the hydrogen. So the covalent ones, that's where the atoms were sharing their electrons. And if they completely gave up their electron to the other one, then that was where they're donating or transferring. That's the ionic bond, right? And then the hydrogen, we might have had the oxygen that was following the electron that has negative charge, and it gave the hydrogen a positive charge. So now that it has a positive charge, it will be attracted to the other thing, negative charge. When it does that, that's and so me explaining this
Seems easy enough for me, right? Because I freaking lecture over it. I literally just did. They literally just heard me say, dare you. Okay? If you, yes, because if you, even if you're just talking to your steering wheel and explaining it to your steering wheel, but putting the effort into really like verbalizing. Science has proven that verbalizing that stuff is the end of the one way to show you actually are learning or to help you learn that stuff. Okay? Because thinking through the way to actually talk about these things, that helps your brain like make sense of it. Instead of just seeing out words you're writing out that I'm spitting at you. Your brain has to make enough sense of it to spit it out of your mouth. And then I can explain it to you that it would make sense. And then you can explain it to somebody else that it makes sense. And they're agreeing, yeah, that makes sense. You probably understand it. That's the idea. Dare you to make the sheet. Dare you to verbalize it. Even if you are like just for whatever reason, just like having that weird, really weird. That's fine. If you can at least think through verbalizing it to somebody, that'll help. It's something. It's better than not. I just really challenge you guys. Make that summary sheet and then explain it. To somebody is the best. Right? To somebody is the best. All right. So that's my recommendation on how to study for this course and literally any other course that you're ever going to take in your life. Being able to explain something is the key to understanding. Okay? Take that time to be able to explain it. It doesn't work for you. That's it. Does anybody ever have any questions about any of that? Anybody have any suggestions for anybody? One thing one of my students in my hybrid class brought up is that on a lot of YouTube videos, it automatically makes closed captions. And you can actually search within those closed captions for terms. So you could like search in my video if you forgot what inoculation was in lecture. And afterwards you realize that, or you took your test and you can remember what it was. I'm gonna go look that back up in the lecture, search for it in the video. And it will find it for you, find that part of the lecture for you. So don't forget that function as well. All right, you guys have everything you need to succeed in this class, you really do. And we'll have our review on Friday at 10 a.m., tomorrow at 10 a.m. Join or don't, I'll still record it and post it, but I hope that you can join. If you have any questions, please bring them to the review.
I know, that's what happened to me.
I
don't get it. Okay, have a good day. See you next week.
All right, so for like study sheets, hold on.
Yeah, it's like I was with her when she was just talking about H2O, and then she like lost it.
Open up here. I'm just wanted to search.
But
I need to print and everything like that, yes. You'll do that.
I'm gonna record it.
That's everything.
Yeah.
Well, thank you.
Tuesday,
This
I'm
time,
having the
everything's
other weekend too.
If you were somebody that wants so we'll have all this information on it, as well as the rest of the chapters for unit 1, so 4, 5, and 7. Then you would be able to find that on that.
Have a good weekend.
I'll see ya. Did you have a question?
I did, but I'm gonna watch, just the hydrogen bond thing. So I'm gonna watch a couple videos and then watch the lecture.
Yeah, and then definitely email me
Yeah.
or come to their, or on the review, you can always just like pop in and be like,
Yeah.
hey, what is this? Can you explain it again?
Yeah.
I'm happy to do it. Yeah.
Thank you.
Right, anyways. The top piece we also talk a little bit about like what's going on. So if we're still continuing with 1, and I'm telling you that you can find your big picture study sheet for wherever it's supposed to be. So for unit 1, do a unit 1 review, and it's a unit 1 study sheet. So if you were to click on this, everything that can be on unit 1, and it's the same exact topic that were on your weekly one. It's just all on one sheet. So if you want that all on one place, just have that one sheet, and then you can like make your that's there for you as well. And again, just wanted to by week thing do that too.
So our quizzes are due tomorrow,
Yes, by midnight.
and then how do you go to the 5?
That is Just look back. We'll go back to. Scroll a little bit. You're okay. So we should
One.
and you'll get used to navigating. Yes, you'll get used to navigating as we go. Take your time, figure it out. Slides will be in your section. Overview, you'll find your study sheet, what you should learn, review, extra stuff to stuff for the lecture. The lecture materials will be in long. It'll tell you to lectures, labs, and tools, all that. To find with chapter 1, that's chapter 1, 3 slides. And then chapter 2, chapter 2 slides. So you will click on these guys.
That's where I'm confused, like I clicked on it when we were doing it, but I didn't know how to use it. You said F1, or like, where you doing it?
So once you have this, you can open these and see in Canvas, but again, download it, right? So you click that. And went down to what's possible. Download for Windows computers. So you can just click on it to open the file. Okay. Always have some sort of pop-up or something, right? And you can click enable it, and then you'll open up the links themselves that are in it. That way you will access to it. And then you'll have just control F. And you'll automatically find one, you can type whatever term you're looking for. That help? Yeah. Awesome. If you get lost on anything else, just, come and see me in my office hours or email me. You know, some screenshots if you're not sure. Okay, I'll be there to help you.
So pretty much if we follow that study sheet and, I'm gonna try and read the chapters, of course, but if we do the study sheet and do the slides, we should be okay with the quizzes and the tests.
Yes.